Hello everybody and welcome to today's webinar. This is the third webinar of a series of 3 where we have been looking at different aspects of cell death. In this session I will talk to you about the different types of cell death that has been described up to date and how we can differentiate between them. As I've mentioned, in this session we will look at different types of caspase-dependent and independent cell death and what are the key markers for each type. So let's have a recap about cell death and the different types. What is cell death? Cell death is an essential mechanism that takes place in multicellular organisms. And we know of its importance because deregulation of cell death is at the basis to multiple disorders. If cell death occurs when it's not necessary, it can lead to pathologies such as type 1 diabetes, by excessive death of the pancreatic beta cells, or autoimmune diseases like rheumatoid arthritis. On the other hand, if cell death is not active when it should, it can lead to hyperproliferative conditions, of which cancer is the best known example. And why is it important to understand cell death mechanisms? Because, as you probably know, there are multiple ways by which a cell can die. From an experimental mechanistical point of view, we want to know how cell death events are happening. But from a therapeutic point of view, this information can help us to find an appropriate treatment for a disorder. So for example, tumor cells are generally resistant to chemotherapeutic agents that induce apoptosis. So perhaps in these cells, we can use agents that induce cell death by necrosis to kind of get rid of them. And so, before we study how cells are dying, we have to ensure that the cell is dead. And just once more, let me remind you how to clarify when a cell is dead. So according to the NCCD, the Nomenclature Committee on Cell Death, researchers should define a cell death when the following features are observed. The cell has lost the plasma membrane integrity, the cell has undergone complete disintegration, and whatever is left of the cell has been phagocytized by the neighboring cells in vivo. We already looked briefly at the different types of cell death in our previous webinar, but let's have a quick refresh. This is the classical view of the different types of cell death that most of us are familiar with. We talk of apoptosis as a specific type of programmed cell death, whereas necrosis is considered non-programmed cell death. Well, as we mentioned previously, this view is no longer that simple. The Nomenclature Committee of Cell Death suggests that cell death types can fit into 2 mutually exclusive subtypes. Accidental cell death does not involve molecular machinery and cannot be modulated. This type is unlikely to happen in vivo and reflects more the conditions that in vitro cells can be exposed to, like being frozen improperly. The type of cell death we all have in mind falls within the regulated cell death, which involves a molecular machinery and therefore can be modulated. And within regulated cell death, we find programmed cell death, which happens as part of the development program or to preserve tissue homeostasis. So here you can see an overview of all the different types of regulated cell death that have been described so far according to the NCCD. So you can see that we don't talk nowadays of just apoptosis and necrosis, but that we differentiate between non-programmed and programmed cell death, and that within programmed cell death there is a broad range of cell death types. So let's look at programmed versus non-programmed. As we just mentioned, necrosis is a regulated but not programmed cell death mechanism. Necrosis is generally defined by a lack of programmed cell death features. This means that if dying cells do not show apoptotic or any of the other type of cell death features, then they are considered necrotic. But necrotic cells do have some defining morphological and biochemical features, and some of them are, for instance, no ATP is required to trigger necrosis. In vivo conditions, the release of the cellular content can cause inflammation of the surrounding tissues. And necrotic cells show swelling following by karyolysis, which is the dissolution of the chromatin and the nucleus. There are no assays to really look at necrosis per se, but there are few markers that can be used to define cell death and necrotic. However, they have to be used in the right context. For instance, release of LDH, lactate dehydrogenase, can be considered as a necrotic marker, as the lysis of the cell will lead to the release of the cytoplasmic content into the culture medium. So as you can see on the image on the left, where we have measured release of LDH into the medium, and then broken down cells will give a higher signal than healthy cells. As we discussed in our apoptosis webinar, analysis of phosphatidylserine exposure, which is considered an apoptotic marker, in conjunction with viability dyes, can be used to discriminate between necrotic and apoptotic cells. So as you can see on the image on the right, we can see how Jurkat cells treated with staurosporin can die by apoptosis, shown in green, or necrosis, shown in red. Remember, however, that timing is very important when we look at phosphatidylserine exposure, as at later stages of cell death, even necrotic cells will show PS exposure. So let's look now at caspase-dependent programmed cell death. Apoptosis is the most well-known caspase-dependent cell type, but caspases can also be involved in other lesser-known types such as Anoikis or Pyroptosis. So let's look at them in a bit more detail to see what are their unique defining features and how we can differentiate between them. And although we devoted our previous webinar to apoptosis, let's just have a quick look at it again. The extrinsic apoptotic pathway is induced by the extracellular signals that are sent and propagated by specific transmembrane receptors such as Fas, CDC95, TRAIL or TNF-alpha. Activation of these receptors leads to activation of caspase-8, which in turn leads to activation of the effector downstream caspases, caspase-3 and 7, to activate proteins that will lead to cell fragmentation. The intrinsic apoptotic pathway, on the other hand, can be triggered by intracellular stress conditions such as DNA damage or oxidative stress, all controlled through the mitochondria. Activation of the pro-apoptotic members of the BCL2 family lead to permeabilization of the mitochondrial membrane, releasing APAF1 in other proteins in order to form the apoptosome, which includes active caspase-9. Active caspase-9 will in turn activate the effector downstream caspases, caspase-3 and caspase-7, to activate proteins that will lead to cell fragmentation. So what defines apoptosis? Historically, apoptosis has been defined by its morphological features, which include rounding up of the cell, nuclear fragmentation, or plasma membrane blebbing. However, as morphological features can be shared by various different types of cell death, as we will see in later slides here, apoptosis is better defined by biochemical features such as activation of pro-apoptotic BCL2 family members, caspase activation, and oligonucleosomal DNA fragmentation. We discussed in detail all these features and methods to detect them in our previous webinar about apoptosis markers. So I would recommend you to look at it if you want to know more about those details about apoptosis. The Apoptosis Markers webinar is available to watch on demand from our webinar library. Right, so we've reviewed apoptosis. Let's now look at other caspase-dependent cell death types. Let's first look at Anoikis. Anoikis is related to apoptosis, and it shares many of apoptotic features such as caspase activation and activation of pro-apoptotic BCL2 family members, BIM and BEAT in particular. In normal conditions, when adherent cells are attached to the extracellular matrix, integrins present in the membrane are responsible for transducing pro-survival signals. When the interaction with the extracellular matrix is lost, this pro-survival signal is inhibited and the intrinsic apoptotic pathway is activated, probably through Bim and Bid accumulation, but this process is not yet clearly understood. Anoikis is important because it prevents the readhesion of cell into new matrices. When Anoikis is not functional, an adherent cell is able to survive in suspension or is able to grow and proliferate in a different location. And this is generally what happens in metastatic conditions. One easy way to assess whether cells are dying by Anoikis is by assessing the cell viability using an encouraged resistant plate. Here you can see how BJ-third cells are able to grow on a normal plate that's on the top section of the image. You can see on the top left side the live cells stained with calcein, which is a live dye. When the cells are grown on a plate that doesn't allow adhesion, like an encouraged resistant plate, the cells start to die. So you can see there is a lack of calcein signal on the bottom left image. The appearance of nuclear condensation, visible by using a specific membrane impermeable dye called ethidium homodimer, confirms that we are looking at Anoikis. So let's look now at pyroptosis. Pyroptosis is an inflammatory caspase-dependent form of cell death that happens after microbial infection. Morphologically, pyroptotic cells look like necrotic cells, but the cell death is driven by inflammatory caspases, caspase-1 mainly, and caspase-4 and 5, or caspase-11 in mice. As it depends on caspase activation, pyroptosis shares some apoptotic features such as DNA fragmentation or nuclear condensation. Pyroptosis is initiated by the activation of canonical or non-canonical inflammasomes. The canonical inflammasome is activated when the node-like receptors detect pathogens and recruit the ASC adapter protein, which leads to the merization and activation of caspase-1. The non-canonical pathway is activated by lipopolysaccharide directly binding and activating caspase-11 in mice or caspase-4 and 5 in humans. Inflammasome activation will then lead to gasdermin D cleavage, which will drive the onset of pyroptosis. So who are the key players to study when trying to look at pyroptosis? Caspase-1, which is involved in canonical inflammasome formation and responsible for cleaving gasdermin D. Caspase-4 and 5 in human or caspase-11 in mice, which are involved in the non-canonical inflammasome formation and responsible for cleaving gasdermin D. And gasdermin D, which is thought to be the executioner of pyroptosis, probably through the secretion of IL-B beta and IL-18, although it is not clear yet how this works. So here are some tools you can use to study pyroptosis. Gasdermin D, the executioner of pyroptosis, is a 53 kilodalton protein that is cleaved in a 43 and a 30 kilodalton band when active. We have recently launched a rabbit monoclonal against gasdermin D, and we have confirmed the specificity of the antibody using a knockout cell line that does not express gasdermin D, as you can see here on the right. Another key protein involved in pyroptosis is caspase-1. Caspase-1 can easily be detected as a 45 kilodalton band in Western blot. Upon activation, mature caspase-1 can then be detected as a 40 kilodalton band. However, as we discussed in our previous webinar, to ensure that cleaved caspase-1 is active, you should perform an activity assay, and here as you can see on the image on the right. So if both apoptosis and pyroptosis are caspase-dependent cell death types, how can we differentiate it? Well, They have quite different morphological and biochemical features, which have been highlighted in green here in this table. I would say that the main distinctive features are that Pyroptosis displays a lytic and inflammatory morphology in vivo, that the effector caspases, caspase 3, 7, and 6, are not involved in Pyroptosis, and that PARP cleavage is not really observed in Pyroptosis. So, and as we have discussed previously, to be able to define the type of cell death, we need to analyze multiple markers, because as we've seen, some of them can be shared between different types. So let's now look at caspase-independent cell death types. So necroptosis and pyroptosis are the most well-known mechanisms of caspase-independent cell death, but there are other minor pathways such as entosis or netosis, which have been described recently. So let's go through some of these in more detail. So let's talk about necroptosis first. Necroptosis is a caspase-independent form of programmed necrosis. Morphologically, necroptotic cells look like necrotic cells. The most defining feature of necroptosis is the activation of the RIP kinases, specifically RIP kinase 3, and the pseudokinase MLKL. Necroptosis is inhibited by caspase-8. So in the majority of scenarios, cells will choose to die through apoptosis through caspase-8 activation. However, some pathogens are able to inhibit caspase-8 activation, and in these cases, necroptosis is an important backup system to destroy these cells. The death receptors that are involved in activation of the extrinsic pathway are responsible for necroptosis activation as well. So in normal conditions, the death domain of the receptors recruits the pro-survival complex 1, which involves a polyubiquitinated RIP kinase 1. When RIP kinase 1 is deubiquitinated and caspase-8 is active, a complex called complex 2A is formed, leading to activation of apoptosis. If, however, caspase-8 has been inhibited, another complex called complex 2B is formed. Complex 2B recruits and activates RIP kinase 3, which in turn phosphorylates MLKL in order to trigger the necroptotic pathway. In some specific circumstances, activation of necroptosis can occur independently of death receptors, be it toll-like receptors, TLR, or viral infections. Activation of toll-like receptor 3 or 4 recruits the adaptor protein TRIF, which forms a complex with RIP kinase 3 and then activates MLKL. Viral DNA can be recognized by DIA, which stands for DNA-dependent activation of interferon regulatory factors. DIA can recruit directly RIP kinase 3 in order to activate MLKL. So, who are the key players to analyze to investigate whether necrosis is involved in our type of cell death? The RIP kinases, RIP kinase 1 and specifically RIP kinase 3, which are involved in necrosome recruitment, phosphorylated MLKL, which is the mediator of the cell death, and caspase-8 because it inhibits necroptosis. Other minor players are the ubiquitinase CYLD and ubiquitin ligases CIP1 and CIP2. So here are some tools you can use to study necroptosis. MLKL activation can be studied using an antibody against phosphorylated serine 358, which is a residue phosphorylated by RIP kinase 3 and is necessary for MLKL dimerization and activation. So here we treated HT29 cells, which is a human colorectal adenocarcinoma cell line, with TNF-alpha, a SMIC mimetic molecule, and CFAT, as this treatment has been shown to induce MLKL activation. On the bottom section, you can see another phosphospecific antibody, which is detecting, in this case, phosphorylated serine 227 on RIP kinase 3. We have also induced RIP kinase 3 activation with TNF-alpha, SMAC mimetic molecule, and CFAT. And also, I wanted to show you this blot in particular to demonstrate that we also get dirty blots, and some antibodies give extra bands no matter how purified they are. So now that we have learned about necroptosis, the main caspase-independent cell death pathway, let's briefly describe some other minor pathways. So pyroptosis is another form of cell death which is independent on caspases. The morphology of pyroptotic cells is actually quite different from both apoptotic or necrotic cells. The main defining morphologic feature seems to be that mitochondria are smaller with increased membrane density. Also in pyroptotic cells, there is a lack of plasma membrane rupture or blebbling, and there is no nuclear condensation. Pyroptosis is characterized by the accumulation of lipid peroxidation products and reactive oxygen species derived from ion metabolism. Pyroptosis can be triggered by inhibiting the system Xc-, responsible for maintaining the redox homeostasis by importing cysteine, which is reduced to cysteine and used to synthesize glutathione, the major antioxidant molecule in cytosol. Depletion of glutathione peroxidase 4 has also been shown to induce pyroptosis, as the lipid peroxide cannot be reduced. So the main markers to look at induction of pyroptosis are glutathione, the main antioxidant molecule in the cell, and glutathione peroxidase 4. Inhibition of any of them can induce pyroptosis. I will briefly talk about autophagy or autophagic cell death. Autophagy is characterized by the sequestration of cytoplasmic material in vesicles called autophagosomes, where they will be degraded by lysosomal enzymes. Initially, autophagy was described as cell death pathway type 2, and it's true that in specific cases, autophagy can be involved in cell death. But it is thought nowadays that in the majority of scenarios, autophagy seems to promote cell survival and not cell death. And because of this, and because it's a whole topic in itself, autophagy won't be discussed further in this webinar. But if you would like to know more about autophagy, please have a look at our autophagy guide available for download at appcam.com/autophagy ebook. And then there are other lesser known or well-studied pathways, such as the ones you can see in the list. Pathanatos is a regulated necrosis characterized by overactivation of PARP and ATP depletion. Necrosis is a pathogen-induced cell death, specifically in neutrophils, characterized by the release of a chromatin structures called nets. And ptosis happens when a cell invades another one, and this has been called cell cannibalism. It is characterized by activation of the Rho GTPase and its downstream effector ROCK. So now that we have reviewed an extensive list of multiple types of cell death, we want to know, of course, how do we know which one is happening in our system? So there are few key questions and issues to consider before you start thinking about starting lots and lots of markers. So for instance, what exactly is triggering cell death in our model? A microbiological pathogen is more likely to activate cell death via death receptors than by directly inhibiting molecules, which is a mechanism of action that we'll probably see in cells treated with some chemical compounds. So this can help us to already have an idea of which type of cell is more likely to be triggered. Another key issue to look at are the morphological features displayed by the cells in vitro and in vivo. Many of the features described here, like inflammation, won't be seen in vitro cultures, But there are other features like obvious chromatin condensation that can help us to discard which death type is not happening. One of the most useful questions to investigate is whether the death is affected by a certain compound. We'll look at this in the next slide. By chemical inhibition or genetic inhibition, if available, can help us to investigate which molecules and proteins are involved in the cell death, and then we can be able to specify the type of cell death that we're looking at. So following this type of key questions, let's see how we can try to pinpoint which type of cell death is happening in our system. So if, for instance, we look at our cells in the microscope and we see that they are shrinking and not exploding, we're probably looking at apoptosis. So in this case, we should look at a range of apoptotic markers for confirmation, just as we discussed in the previous webinar. If we think that we are looking at Anoikis, perhaps because there is Bim overexpression or the cell line we are using is known to metastasize, we might want then to test if the cells can survive in an encouraged resistant plate. If we look at our cells and then there's some swelling and lysis, then we're probably looking at some other type of regulated necrosis. If our cell type that we're using is a neutrophil, we could be looking at netosis. If the cell death is prevented by CYVAD and FMK, which is a caspase-1 inhibitor, then we are likely to be looking at pyroptosis. If the cell death is prevented by necrostatin, which is a RIP kinase 1 inhibitor, then it's likely to be necroptosis. If the cell death is prevented by ferropstatin, which is a lipid rose scavenger, then it's likely to be ferroptosis, and so on and so on. And if cell death can be prevented by any compound we test, then we might actually be looking at necrosis. So please keep in mind that this is just one example of the multiple ways you could use to analyze what type of cell death you're looking at. Because in most experimental cases, you are likely to already have an idea of what is happening in your model. So you're more likely to want to differentiate between just two potential cell death pathways and not 5 or 6 like we're showing here. Because remember, just because all these pathways have been described, it doesn't mean that they all can be triggered in all cell models. And of course, in each case, once we think we're looking at a cell type, we still have to look at the key markers for each of them. So in this table, we have tried to summarize the defining characteristics of each of the main cell death pathways that have been described up to date. Apoptosis can be triggered by death receptor activation or intracellular signals, whereas pyroptosis is triggered by pathogens activating the inflammasomes. Similar to the extrinsic apoptotic pathway, necroptosis can be triggered also by death receptor activation, but only when caspase-8 is inhibited. Pyroptosis is triggered by ion-dependent lipid peroxides. Necrosis is a toxicity-induced cell death and does not require any specific triggering stimuli. All these types of cell deaths, except apoptosis, can generate an inflammatory response in vivo. Initiator caspases and BCL2 family proteins are the key defining proteins involved in triggering apoptosis. Other caspases, in this case the inflammatory caspases, together with gasdermin D, are involved in pyroptosis. The RIP kinases 1 and 3 and the pseudokinase MLKL are responsible for the onset of necroptosis. Inhibition of glutathione peroxidase 4 and glutathione leads to activation of ferroptosis. The executioner caspases, caspase 3 and 7 mainly, are responsible for executing apoptosis, whereas interleukin 1 beta and interleukin 18 are thought to execute pyroptosis, although as we mentioned, this pathway is still not very well defined. How necroptosis is executed is still unclear. And an increase in cellular reactive oxygen species and decrease in glutathione pool seems to be involved in the later stages of ferroptosis. Knowing which compounds inhibit each mechanisms of cell death is a very useful tool, as we have seen in the previous slide, since we can use it to analyze which type of cell death we're looking at. Apoptosis can be inhibited by CFAT-MK or QVD, both well-known broad caspase inhibitors. Caspase-1 inhibitor, CYVAD-FMK, is commonly used to inhibit pyroptosis. Necroptosis can be inhibited by addition of necrostatin-1, which is a RIP kinase 1 inhibitor, or nicosulfonamide, an MLKL inhibitor. And pyroptosis can be inhibited by ferrostatin-1 or leproxstatin-1. And although morphology should not be used to differentiate between the different types of cell death, it still gives a valuable information for us when used in context. Apoptotic cells show a quite different morphology from the other cell types, such as cell rounding, plasma blebbling, and formation of apoptotic bodies. Pyroptosis and necroptosis share some morphological features, such as the rupture of the plasma membrane and release of cell content. Pyroptotic cells have a very distinct morphology, such as smaller mitochondria, which is a feature that is not seen in any other cell type. So, as we have seen, cell death is a complex business. And because what often happens in life systems is that cells that are in the same tissue or culture conditions, and even if they are treated the same way, they can die by different mechanisms. And this is because cell death can be influenced by several things. The type of a stimulus, for instance. Different types of stimuli are more likely to activate some types of cell death than others. The cell type. Some cell types are more likely to activate some cell death pathways, like for instance, necrosis is only seen in neutrophils. The cell environment. In an in vivo environment, for example, cells closer to a blood vessel will hit a higher concentration of a pharmacological compound than the ones farther away, and this could lead to activation of different cell death pathways. And the cell physiological state. A cell that is entering mitosis will probably respond differently than one that has just finished mitosis. And a cell that has mutations might activate different pathways as well. So this brings us to the end of the webinar with few take-home messages. Remember that cells can die by a spectrum of cell death types, even under the same stimulus or on the same tissue or cell culture. Each cell type can be defined by particular morphological and biochemical features. And although those morphological features can help to suggest what type of cell death is happening, the underlying biochemical mechanisms need to be studied and confirmed. Make sure that you use activators and especially inhibitors of the different pathways in order to confirm or discard what type of cell death you are studying. Apoptosis is the most studied form of programmed cell death for a reason. So if in doubt, I would suggest to first confirm or discard apoptosis in your model before you start looking into other types of cell death. And as we have repeated through the series of this webinar, always take your experimental model and settings in consideration before you draw any conclusions. So thank you very much for your attention. I am ready now to answer any questions. Yes, so one first question is, what is the best way to detect pyroptosis and necroptosis in situ? So I assume by in situ you mean directly in a tissue. So that's actually the best way because you can see how it's happening in an in vivo model. So obviously you might have some ideas. Looking in situ might give you some information about whether there's inflammation happening. Another way it could be using antibodies against the active proteins. So, for instance, for necroptosis, you could use the phosphor-specific MLKL antibody. So yeah, I would say probably morphology in combination with some immunohistochemistry staining. That would be one of the best ways. Another question is, can an apoptotic cells express BCL2 on immunohistochemistry, for example, a tumor cell? Yes. I mean, BCL2, any of the BCL2 family members is probably expressed in one level or another. What is going to happen in an apoptotic cells is that the balance between the BCL2 molecules and the level of pro-apoptotic members, such as BAX-BAT, is on the favor of the pro-apoptotic members. So you could detect some BCL2 expression. You might want to compare it maybe with a treated control and see what's the level in an untreated control. So that might give you an idea whether the staining is just a basal level or actually there's something happening in your model. Another question is, Anoikis can also be called apoptosis. For example, what if Anoikis is also positive for tanol? As we mentioned, an Anoikis is a specific type sort of apoptosis. So what is likely to happen, that a lot of the markers that you see for apoptosis, pretty much all of them, I would say, appear in Anoikis. When Anoikis is different, it's because it has to do with the cell environment. So what is going to happen is that because your cell is able to survive in suspension, whereas a normal cell will just die. That's the way you can detect it. It's also tricky when we look at our cells in culture, because most of the cells that we all have in culture are some sort of tumor cell in a sort. So they are likely that they could survive in a, they could have their Anoikis pathway inhibited. So you could see that. Probably the best way to see if in your cell model Anoikis is inhibited is by using the encourage plate, because that will tell you that actually that cell type is able to grow even when encourage is not needed. So that could give you that sort of information. But it's very likely that you're going to see very, very similar markers as apoptosis, because at the end of the day, apoptosis, Anoikis is a sort of a subset of apoptosis. It's just apoptosis happening in a very specific model. Another question is, are these cell death mechanisms mutually exclusive, or can they coexist? It depends what you what you mean by mutually exclusive. What happens, I think, in one particular cell, probably only one of them is happening. But of course, you're not looking at one individual cell at a time. Usually when we do the experiments, we look in a cell culture in a tissue context. And then what happens is that you might see different types of cell death happening in the same culture, as we said. And also, if you remember when we were talking about necrosis, and I showed the apoptosis necrosis kit, on the same cell culture, we were seeing apoptosis and necrosis. So yes, they can happen. Some of them can happen in the same culture, but not on the same cell. And that's going to depend, again, on the type of your cell, on the stimuli. So you have to take that into context. For instance, some cells, they will behave differently when they're exposed to low concentrations of a drug, and they will trigger different type of cell death when they're exposed to high concentration. So that's something you have to keep in mind. I guess as a scientist, the best way we can say is, well, in the majority of my cell model, this is the type death that is happening, and therefore you could be quite confident that that's the cell type you're looking at. A question is, regulated in a sense comes under program, don't you think? Well, yes, it is. The difference, I think, when we talk about program, what we're saying is that there is some specific, there's a trigger that will make that cell death pathway happening. And I think when people try between program and non-program, is that, of course, necrosis is regulated when the tissue, the body makes some cells or tissue go into necrosis, it's because something it's happening and it's necessary for that tissue to go into necrosis. The main difference between the program is that the program tends to really refer to very specific cell types, and there's some stimuli required. But I agree with you, maybe something that is more a semantic difference than actually the difference in function. Does cell death varitypes depend on the cell cycle phase on the cell? It might do. Again, as I said, a cell that is entering in mitosis will have different proteins activated than a one that has just gone through. So there will be different checkpoints in action. P53 might be involved, and P53 we know sometimes might be involved in blocking apoptosis. So it might depend. Again, in vivo and in our cells, it's all a living system. So you have to look into the context of where the majority of the cells are happening and what's going on over a period of time. Another question is how to differentiate between Anoikis and apoptosis by assays and markers. I believe I have answered those questions before. As I said, apoptosis and Anoikis are kind of the same. The main difference is Anoikis happens when a cell is not able to survive on suspension. But if you believe I haven't answered that question, please let me know and I'll try to get through that again. Do you recommend a particular agent inducing for Anoikis? Because, as I say, Anoikis in a way is apoptosis. I presume it will depend on your type of cell. Anoikis will be induced when a cell that should be attached is not attached. So probably using an anchorage resistant plate would be a good way to induce Anoikis. Does the collagen degrade in the fibrotic tissue belong to cell death? And what kind of cell death? Ooh, that is a really good and tricky question at the same time. I really don't know. I would have to do a bit more digging. I would say that the degradation of the collagen is a collateral action of the type of cell death. Because I guess what you want to see is that the cells within the collagen matrix, what's happening with them. So you might want to look into two different ways. You might want to look just at your cells outside of that collagen matrix first and see what's happening. And then what's happening around when those cells are with the collagen might be triggering different sensors. I like to think that it's probably some sort of lytic cell death. So it's going to be probably a kind of caspase-independent, just because caspase-independent cell death tend to release sort of the cell content, and that tends to create a sort of lytic mode around the tissue. So another question says, if I'm not grown, it has been reported necrosis-like morphology of cell death with caspase-8 and caspase-3 involved. What would that be then? I think that is more likely to be necroptosis, because the downstream pathway of necroptosis are still not clear, so I would not discard the fact that maybe there's some caspase-3. And again, it comes back to what I was saying, that it depends if you mean there's caspase-3 in the culture or there's caspase-3 in the cell. So it could be that it's very likely that on the same cell culture you're seeing different types of cell death. Another question is, can we use ATP levels and LDH release to differentiate between apoptosis and necrosis? You could look at that initially, just to give maybe a quick idea, am I looking at this, am I looking at that? But if you wanted to make sure that you're looking at apoptosis, then you have to look at some other apoptotic markers. But definitely, if what you're seeing is maybe no changes on the ATP levels and LDH release at early stages, because obviously at some point later on all the cells are going to die and the whole culture is going to go horribly long, so you're likely to have a lot of LDH in the culture regardless, then that could give one idea. Another question is, how can I prove caspase-independent cell death? Well, you can use the caspase inhibitors, like we say CFAT and the QVD, and if then you don't see any effect, then it's quite likely that you're not going to see the cell death that you're looking at is caspase-independent. Another question is, when I analyzed tissue prepared from diseased animal models, I saw many pynotic cells, but many of them are negative for tunnel and cleave caspase-3 by IHC. What is your recommendation? Oof, a good question. For that one, if it's okay with you, I will follow up with you offline, because then I have time to look through my notes and look in more detail what type you're looking at. So if that's okay, I'll follow that question offline. Does cell death mechanism change from one to another? For example, cells die by necroptosis, which is spreads dead cells to neighboring cells by pyroptosis, is due to damp from necroptotic cells that can trigger inflammation. That is quite a model indeed. To be honest, the question is, I don't know. I think it could change slightly. I don't think it's likely that you're going to see a huge change from a necroptosis, so from a caspase-independent to then a caspase-dependent to then to a necrotic. What I think it would happen is that the surrounding cells around that initial cell are going to have very similar type of cell death. And then if the stimuli that it's provoking that cell death is not passed to the other cells, some of them might be affected by some of the dams, some of them might not. So, and actually that follows quite good to the next question. Someone saying, I have heard of DAMPs. Could you tell me what they are? So DAMP stands for damage-associated molecular patterns. So these are molecules that can lead to, for instance, an inflammatory response if there's no infection. So it's what we see in necrosis and necroptosis. So that's kind of what it is.